So this is something that is, it's hard to approach this without some degree of emotion, because this is something that I have been a part of and watching in some fashion for about 20 years. So I wanted to say where, what have I seen, and what have I seen coming toward us in the future. And I really have to thank the organizers for putting this together. This is a long time in the making. When I first started with this, there was effectively nothing about this condition except for a couple of papers that were coming from Japan and I think one from Korea. So it's the additional information over the past 20 years has just been astounding. I wanted to go back to the original paper, or the family that I saw, and to put a face on it as I saw it. And these two boys are the original children that I saw. When I saw them, they did not look like this. They both had come down with fevers and upper respiratory infections. So they had fevers 103. They were bouncing back and forth between the emergency room for a number of days because they were saying, as Kim Smith had noted, bad things happened in their family and nobody seemed to know anything about it. Well, they finally ended up at a state of coma. Both of them were having seizures and signs of encephalopathy. And the MRI of one of the boys looked like this, with symmetric lesions in the thalamus and other parts of the brain, usually involving gray matter. And it was a fairly specific distribution of lesions, and nearly identical between these two boys who happened to be twins. This was a very unusual presentation, and one of the confounding factors was the grandfather of these twins came in to say, "It's Lee syndrome. I know that because I had it as a kid, but I got over it." Well, Lee syndrome is a mitochondrial disorder, and it's a very unusual statement for somebody to say, "I got over it." So that really kind of tripped some alarms to say this is something a little bit different. But these children looked so bad that they were extubated with the idea that these children were going to die. They looked, everyone thought that this is where it was going to go. But this is not a before-after picture, this is an after picture. So these children woke up, and they woke up having lost a majority of their functional skills, and it took them about 4 months to regain them. This picture was actually taken about 9 months later. And so they are now back at functional level at the time this picture was taken. Although I'd love to say that this was the end of the story, it was not. Unfortunately, one of the twins developed a recurrence a year later and died. The other twin after that developed a recurrence and has sustained permanent neurologic damage following. And this is one of the patterns that we've seen throughout this family, is that there have been a combination of recovery, some in full, persistent damage, and death. And this is the pedigree that they belong to. And everyone in blue is a person that has had an acute necrotizing encephalopathy episode, as best we could tell. This goes back some time, including to times at which they did not have MRI scans. So we didn't have information in the same way for everybody. Everybody in purple is a person that was a parent and was in a relative position to those other patients. And then everybody in green were people that we were able to collect for a study. And so using this family structure, we were able to do something that we don't see very much of anymore, but a process called linkage analysis, where we actually did a matching process to figure out the affected segment for all these patients and to be able to sequence all the genes in that region. Doing so, we were able to find that there were variants in the RAMBP2 gene. These were not easy to find, and it was also very difficult to convince people that these were real. I won't get into that story, but it took a while to actually get this out there. What has really been interesting to me over the years is how little variation there is in this gene for the number of patients that we have sequenced. For all the patients that have had RAMBP2, we have only found, my group has only found 3 pathogenic variants. There have been two others that have been published in the literature that are generally accepted as likely to be true also. They both come from single cases. And there's a third report from a different location of this protein that has also been published, which I'll get to in just a minute. What is also interesting is that all of these are missense changes, and they all cluster in a very similar location in what has been called the leucine-rich domain. One of the questions, of course, is how does this all work? And all I can say at this point is one thing that I really don't think is the case is it does not seem to be a loss of function or a null allele. And that's because, one, we don't see the range of premature stop codons or frame shifts that usually characterize loss of function alleles. The other is that we have had a number of patients referred or to me directly that other physicians have wondered, we have a patient who has a deletion across this region and the RAMBP2 gene is gone. Will this child get acute necrotizing encephalopathy? And to date, none of those children that have been sent have actually had it already, and subsequently I've not had any follow-up reports suggesting that this does occur. The other thing that we did, which goes into that giant journal of unpublished research because it just didn't work, is that we actually took heterozygous knockout mice and we infected them. We used 2 different strains of mouse-adapted influenza, and we actually backbred the knockout onto a different strain to see if we could ever make them sick. And the answer was no more sick than the other mice, than their litter mates. And so we were using intranasal inoculation of this mouse-adapted influenza with trying to use the route that had the closest proximity to the brain, and nothing really seemed to happen. So with that, part of that study was in preparation for what I had been working on at the time, which was to make a knock-in mouse. And unfortunately, after 2 rounds of making chimeras and trying to get the offspring, we never got the mutation to go to the germline. So we failed on that process, and that's about the time that the money ran out. And so that was when I really had to put my research at the bench level onto the back burner. But before we did that, we were able to do sequencing on a lot of cases. We had actually collected about 120 persons who had had who had had acute necrotizing encephalopathy. This was divided between 80 families, and we divided this into patients that had a family history versus singleton cases, and those that had recurrence either within the families or not. And what we were able to show is that we could find the RAMBP2 mutation, especially in patient families, in cases that had either recurrence or that were familial. But while that's somewhat interesting, what's also, I think, something I would to consider here is that there were 8 families here that had no RAMBP2 change. So this is not the only player out there in terms of producing this disorder. And from my perspective, what I was hoping to find over time was with more sequencing that we might be able to actually find what are the other genes, because especially at the beginning, there was very little known about all the different functional aspects of RAMBP2. And so it was hard to generate a model that you could test to say, now that we know how this works, now we can show how it's dysfunctional when we change it. That was really the hardest part. The problem was that sequencing really was not very helpful either. So my question is, where are those other genes? So we have actually taken a number of our families and run them through exome sequencing. We were able to scrounge up some pilot funding and ran trios and did not find any good candidates within that sample. And this has also been published from other labs who have been running exomes on patients with A&E, that they are finding variants in RAMBP2. Some of them are outside of the areas I typically think of as the regular area. And some have had possible digenic inheritance with variants in CPT2, which is carnitine palmitoyl transferase. There is a well-known thermolabile variant, especially in Japan, which predisposes to the condition influenza encephalopathy. That's an influenza-triggered condition in which patients have encephalopathy, but really no other MRI changes except for maybe some generalized swelling. And the Japanese groups have published that there are RAM-BP2 pathogenic variants in combination with CP2 variants in their patients. But that's about it when it comes to what else is out there, except for maybe one. And that's what I find really interesting. This was actually just recently published. The nice part about being in genetics is that everyone kind of throws these papers at me the second they come out and available. And these are two different cases of acute necrotizing encephalopathy, or at least A&E-like conditions in patients with microcephaly, little bars in the way of my thing, osteodysplastic primordial dwarfism. So this is a condition where the patients are very small, have a very small head. There's disabilities and things like this. And into the children that have had this, they have had episodes of acute decompensation and coma following infections. The MRIs that I'm showing here are from a child that this first episode was triggered by unknown cause. This second episode was triggered by influenza B. And the other case, it was triggered by human herpes virus 6, which is another strong player in terms of triggering. And so what's really interesting about this disorder is it's caused by autosomal recessive changes in RNU4-ATAC. And so this is U4-ATAC, which is a component of the spliceosome. And so this is actually a non-coding RNA. And this just kills me because you cannot find these on exome studies. And what it really kind of says to me is, to be complete in the evaluation, future studies may really need to consider doing whole genome and just skip over exome entirely. As Kim Smith pointed out, one of the issues that we have with the study of acute necrotizing encephalopathy is how widespread the patients are and the problem of recognition. And so when I am called because there's a child out there that has acute necrotizing encephalopathy, what usually the story is, 5 days ago, we started having fever. 3 days ago, a child came here, and guess what? We've been looking like crazy, and our fellow just found something in the literature and has been reading about it and wanted to talk to you. So most people don't really understand or recognize what this is. And by the time they actually get around to making the diagnosis or thinking the diagnosis, often it's too late to do anything. It's too late to intervene. It's also probably too late to get the samples that you need to establish what is actually going on. That is part of the reason there's such a paucity of data is by the time people think about it, those cytokines, if they were there, are probably gone. And so the obtaining uniform studies has been nearly impossible just from the wide distribution of pediatric hospitals across the United States. So at Phoenix Children's, when I got here, we quickly had about 5 cases within a couple of years. And I think one of the interesting things about our hospital is it's the main children's hospital for a city of about 5 million people, and for most of the rest of the state, which is about 7 million. So most cases really come to our direction. So we had the opportunity to actually catch them early. And so there's been a number of fellows or colleagues at Phoenix Children's that have been interested in promoting more of a dedicated pathway, and this is what it looks like. We published this as a part of a recent paper. And I'll just point out a few things. I'm not going to walk you through everything, but really what we're looking at is whenever we get a child who is having a rapid deterioration in their consciousness in the setting of a febrile illness, we really want to jump quickly to rapid imaging. And here we can do a rapid sequence or a fast sequence MRI. It's a very dirty kind of scan in terms of it doesn't give you the fine details, but it can really show you those hyperintense lesions that come about from acute necrotizing encephalopathy. And if you can't do that, then get them into the CT scanner as soon as possible, so that we can get a sense of whether we do have these bilateral thalamic lesions. And yes, along the way, we have to look at all the other kinds of things, but what we really want to emphasize is getting that early diagnosis and intervention. So obviously, we got to do a lot to stabilize. We want to have a broader workup of CSF, a broader workup of blood, as well as EEG monitoring. And what the paper that we had published was that the presence of sleep spindles was actually seemed to be a protective biomarker of patients. They seemed to do better when those were still present. And then finally, really start treatment right away. And we've talked a little bit already about steroids. We have also been encouraged by the studies of IL-6 inhibitors. So we have encouraged giving IV tocilizumab also once you've gotten your LP and your blood studies to make sure that we've got those cytokine profiles. But to get those on board right away. We have had the opportunity to use this twice now. The results, at the very least, the hardest part, of course, with doing interventional trials with RAMBP2 is that you have no control group, except for perhaps those historical controls. And what we know is that about a third of patients will wake up spontaneously. So when you give a drug and they wake up, do you know that you actually did anything? But at the very least, what you can say is it did not seem to harm or accelerate the process in any way. So at least we are not creating more of a problem in doing this. And just anecdotally, one of the children that received this had one of the MRIs where I was expecting that this child would be awake in about a week. It looked very, very bad. And the child was already waking up when I walked in the room on that very first day. So it was pretty interesting, but again, anecdotal, very early. Okay, now since this is a workshop, I get to ask a couple of questions of the group. So I've been thinking about this whole protein and this disorder for a long time. And I have to ask questions. One is, what the heck is RAMBP2 overall? It has so many different functional domains. It's present and active in so many different processes within the cell. Is this really like a Swiss Army knife where it's really just a connection between lots of different tools that are used at different times? Or is there a greater sort of overriding role in the cell that we could abstract? For example, with its connections to microtubules and motors and packaging components and protective and chaperone kind of functions, is it more like a moving truck? In fact, like a door-to-door packaging company that moves packages from one area of the cell to the other in order to get into and out of the nucleus eventually. And the other is it really just one protein? So we always sort of think of this big, long chain, but does it actually get cut up into smaller pieces? Or do we have alternating splicing? Do we use different cassettes? And are those used at different times in different cells? Anybody that has looked at the antibody staining profiles of the different antibodies for RAMBP2 out there will notice that there are many different bands that light up when you use that. And the 358 kilodalton band is usually not the strongest one. So my question is, are those other bands really just degradation products, or are they actually functional components that are acting semi-independently of the others? The other question that I have is, what's doing this all? What is the most relevant cell? Usually when we're talking about neurologic disorders, we sort of blame the neuron. We go right to that and say, okay, the neuron is somehow falling apart and dysfunctional on its own basis. But one of the really unusual facets about this condition is its predilection for certain areas of the brain. And when you look at it in axial cross-section, it looks just like these parts all look fairly separate. But when you look at it in these coronal sections, you can actually see all of these really seem to be contiguous spread. And where that contiguous spread is coming off of really seems to be elements of the posterior circulation. And so that comes up with the idea, well, is this actually the blood vessels? Is it the endothelial cells where the dysfunction is most manifested? Or is it really housed in the immune cells that are the initial responders to the infection? And then finally, the other cell that's often implicated in neurologic disorders is it the glial cells, which can also mount immune responses. So just a few things to think about as we all move forward with research is that, as you know, the biggest difficulty that I've encountered in trying to model this is the idea that it takes multiple parts. It takes an infection. It takes but perhaps a state of the brain that is more susceptible. It may require certain levels of proteins to be present, cytokines or what have you. And so trying to model that really makes a much more difficult prospect. And when we do our cell culture models, if we do it at one cell, is that really the right cell to be examining? Is that the cell that's really going to be causing the disease? Or is it sort of affected, but not really the driving factor? So hopefully those will be some stimulating thoughts that people can think about, and you can get back to me in another 20 years when you figured it out. Now, hopefully it won't be that long. And I really have to say, I'm so thankful that there are so many people here and so many people working on this protein, because what I would really love to see is the answer to the question, what do we do about this?
Hi, my name is Ming Lim. I'm a children's neurologist at the Evelina London Children's Hospital. It is great to be part of this journey with you all to understand and ultimately conquer A&E. Many of you here today would have lost loved ones. Many have children suffering from this condition. And of course, a bigger group of you who have helped in beginning this journey to support clinical and basic science research in this field. As a clinician, A&E, acute necrotizing encephalitis, is one of the most devastating conditions that I see. Many children are previously normal and healthy and struck down by this condition. It often starts with a simple cold. Children may feel unwell, fever, have a cough, but unlike most colds, it does not get better after 2 to 3 days. By contrast, children get significantly worse and often become comatose after that. This, in most cases, is due to the inflammation that is triggered by the infection, causing the brain not to function well. Key to the management of A&E is the early recognition, which is made by imaging. After the diagnosis is made, when the key imaging changes confirm or support the diagnosis, treatment needs to begin. The key treatment is supportive to make sure that every bodily function is maintained, because often when the brain is that inflamed, it stops functioning. And the second key arm of the management is to switch off the inflammation with rapid, fast, and optimal immune treatment. Sometimes, despite optimal recognition and speed of initiating treatment, the outcome is still poor. There are many things we still do not understand, and that is the importance of our journey ahead to find out how we could improve the care and management of children with A&E.
Hi, my name is Theo Morris. I'm a pediatric respirologist and a scientist in Toronto at the Hospital for Sick Children. I wanted to take a few minutes and just explain to you how the research we do in my lab relates to A&E. So there's a few things we don't know about A&E. One thing that's been observed is that A&E is often preceded by an influenza viral infection. But why influenza and not other viruses? So our lab has expertise in respiratory viruses. We also have expertise in airway epithelial cells. And epithelial cells are the cells that line the surface of your lungs and your airways. And we can grow these cells in our lab. We can then infect them with influenza and study the different cell responses. What's important to know is that we can actually get these cells from individual people. So we use a small little brush, and we brush the inside of your nose. And with the cells that we get, we can grow them in our lab after about 5 weeks or so. The brush isn't painful. If you ever eat sushi, it feels a little bit like taking a little bit of extra wasabi. And if you're in Toronto ever and you want to have a nasal brush, let me know. Anyway, we can grow these cells from humans in our lab and then infect them with influenza. What we plan to do is meet children and families who've had A&E and grow cells from them and compare them to cells from people who have not had A&E and don't have any changes in their genes that are associated with A&E. We can then infect them with influenza and study whether there's any difference in the epithelial response in people who've survived A&E versus people who have not. Moreover, our hospital also has a library of influenza viruses dating back a few years. So we can use strains of clinical influenza that may be relevant as well. And in this way, we hope that we can add to the information that's out there and see whether there is an epithelial phenotype, something different about the airways, the lung epithelial cells, that may be important as we try to understand more about A&E and prevent its consequences.
Hi, my name is Alex Palazzo, and I'm a professor of biochemistry here at the University of Toronto. My lab studies aspects of cell biology pertaining to the nuclear pore complex, and we are interested in how the nuclear pore complex participates in various biological activities, and we're particularly interested in one of the components of the nuclear pore complex that is known sometimes as RANBP2 and also as NUP358. So I'll explain to you a little bit about what we know about the nuclear pore and how it interfaces with various cell biological processes and how this may pertain to A&E1. Our cells are divided into two major compartments, the cytoplasm and the nucleus. These two compartments are divided by an envelope called the nuclear envelope. Now, you may remember from your high school biology that most of the DNA in our cells is present in the nucleus. However, it's important that molecules access the nucleus from the cytoplasm, and as such, they must travel between these two compartments. Now, to cross the nuclear envelope, molecules must pass through little pores in the nuclear envelope called nuclear pore complexes. So again, you have this division between the cytoplasm and the nucleus that's affected by the nuclear envelope, and there are perforations within the nuclear envelope that allow molecules to cross from one compartment to the other, and those perforations are occupied by this large complex called the nuclear pore complex. Now, RANBP2 is a protein that sits on the cytoplasmic face of this complex and is present in multiple copies, and as such is involved in many processes having to do with the transport of molecules between the cytoplasm and the nucleus. Using the latest cutting-edge technology, we've been able to have a closer look at how RANBP2 is situated on the cytoplasmic face of the pore. So this is work that was recently published by Andre Holtz's group from Caltech that appeared in Science, and here we get an atomic resolution structure of the nuclear pore and the arrangement of RANBP2 on the face of the pore. And it may be hard to see, but we have about 40 copies of RANBP2 surrounding the exterior of the nuclear pore. And we think that RANBP2 is monitoring what types of proteins, complexes, and perhaps even viruses are coming in and out of the nucleus of our cells.
Hello, my name is Natalie Arel. I'm a research scientist at the CNRS, which is a public research institution in France. I'm originally from the UK. I studied biochemistry at the University of Bristol. I also did my PhD at the University of Bristol on the topic of colorectal cancer research. And I currently head of a lab at the Institute for Infectious Diseases in Montpellier in the south of France. Our lab is interested in the connection between viral infections and a subcategory of immunity called innate immunity. Now, innate immunity is rather different from adaptive immunity, which most people tend to know about, which involves white blood cells, particularly T cells or B cells, that mount a specific response against a pathogen like a virus. And so, for example, if you've heard of antibody responses or T cell memories, that's adaptive immunity. What we work on is innate immunity, and it's quite different from adaptive immunity because it works in all cells of the body. It doesn't need to be exposed to a pathogen beforehand. And while adaptive immunity is triggered within a few days after infection, innate immunity is triggered within minutes. And so we tend to refer to it as the first line of defense against pathogens. Now, all the cells of our body are equipped with sensors, especially cells that are frequently exposed to pathogens like on our skin or on mucosa. And when a virus or a bacteria infects, then these sensors will detect this pathogen and will set up an alarm system. Now, the alarm is detected by the neighboring cells who are made aware of the fact that there is a pathogen in the environment and that they are likely to be infected. And therefore, these cells then set up a defense mechanism which involves the production of chemical molecules called cytokines. Cytokines will then diffuse in the body, what we call systemically, and they will reach all the organs, including the part of the body that is being infected. And they are very powerful at neutralizing the virus or the bacteria. However, they're also dangerous for the host, and it's quite important that when the job is done, then they are eliminated and taken away from the system. Now, in some diseases, this doesn't occur as it should, and the cytokines keep very high levels of expression, and they attack vital organs. And this is what we call the cytokine storm. And typically, for example, during the COVID pandemic, many of the people who were quite sick with SARS-CoV-2 infection were in fact not sick from the viral infection, but from the inflammatory innate immune response that was triggered by the virus. And in fact, patients with long COVID have inflammation that is now almost chronic, even though the pathogen is no longer in their body. And this is a little bit what we believe is occurring in ANE1 patients, that these individuals who get infected, this triggers the innate immune response within minutes. And for reasons that are not quite clear to us at this stage, the cytokines are not cleared from the system, and they keep being produced, and they cause damage, particularly in the brain. Now, ANE1 patients have a mutation in the gene that codes for a protein called RANLP2. Now, this protein is very interesting to us. We've been studying this protein for many, many years because it's an important component of the nuclear pore complex, which is implicated in transport between the cytoplasm and the nucleus, or between the nucleus and the cytoplasm. And we think that these mutations in RANLP2 could completely unbalance the regulation of cytokine production. And that's exactly what we're studying. So what we typically do in the lab, we analyze innate immune cells from the circulating blood, and we compare cells that have the mutation in RANLP2 versus cells that do not. And we challenge them with viruses, and we see how they produce cytokines, to what extent, and how long for. And we really hope that this work will shed light to help us to understand how ANE1 patients suffer from this cytokine storm, and in ways that we might be able to prevent this.
I wasn't expected to live, and I had to relearn how to walk and do everything I could do before. I really struggle with processing, and I have to work probably 2 times as hard to get the same outcome.
I have the gene of A&E, but I haven't been affected by it yet. And what research would mean to me would be that me and my family can be more cautious in the future and to be prepared. It's really scary for me not knowing what will happen in the future. So research will clear a bit of the unknown of what causes it and how we can be more careful further on in the future.
Research into A&E will mean a lot for me as a mother of, I've got 4 children, 2 that have had recurrent episodes of A&E, and then I've got 2 that have the gene that makes them susceptible for A&E, and so does my husband. So that is quite scary as a mother to have so much unknown about this condition. Some research into it would mean that I could prepare for the future in terms of any recurrent episodes, treatment being more efficient, but also protecting the children that haven't had an episode yet. That's a scary thing hanging over them as well as me. And right through into the future, thinking of when they get into relationships and look at having children, what's the future there? So it's there's so many things. It's so hard when there's so little known about it. So this research would mean the world to me.